Hello, my name is Amanda Price. I'm a program officer in the Division of Neuroscience and Basic Behavioral Science in NIMH, and I'll be emceeing this portion of the workshop. So the objectives of this first session on data analysis, annotation, and integration are to explore challenges in annotating and integrating data sets across individual labs, species, ages, and data modalities, how to characterize brain cell types and states, establish consensus reference cell atlases and unified cell taxonomies, and how to leverage AI and machine learning to improve data integration and spatial mapping, and identify gene regulatory programs linking genetic variation to cellular phenotypes. So our first keynote will be delivered by Dr. Lein and Hongkui Zeng from the Allen Institute. Dr. Lein has led the creation of large-scale anatomical, cellular, and gene expression atlases of the adult and developing mammalian brain, including the inaugural Allen Mouse Brain Atlas, and a range of developmental and adult human and non-human primate brain atlases. His current research uses single-cell genomics to understand brain cellular organization, define cellular vulnerability and disease, and identify regulatory elements that allow cell type-specific targeting and manipulation. And Dr. Hongkui Zeng has led the generation of large-scale access data sets and tools to accelerate neuroscience discovery. She has also led the Transgenic Technology Program, the Human Cortex Gene Survey Project, the Allen Mouse Brain Connectivity Atlas Project, and the Mouse Cell Types and Connectivity Program. And her current research uses a combined molecular, anatomical, and physiological approach to unravel mechanisms of brain circuitry and potential means for treating brain diseases. So with that, I'll hand it off to Dr. Lein and Dr. Zeng.
Well, good morning, everyone. I'm really pleased with my colleague Hongkui Zeng to have a chance to set the stage for this meeting by describing our work in BICAN on cell atlases of the adult and developing mammalian brain. And we really want to have the chance to lay out what's already happening here, especially because so many people here maybe are not part of BICAN and not aware of what's happening, and what kinds of data are we generated and what kinds of integration approaches we're taking to try to create a foundational reference, as Yang was noting. I also really want to note out of the gate that we'll be representing the work of a huge number of individuals here. This has been an extraordinarily collaborative consortium and successful because so many people are deeply engaged in making it a success. So really the North Star that we've been following for BICAN is to try to create this foundational reference for neuroscience and medicine that we'll be primarily describing as a set of molecular cell atlases or cell taxonomies that can describe the cellular makeup of the brain and its molecular underpinnings. However, this is intended to be much more than something to serve the single-cell genomics community. This is meant to be a foundational atlas that serves the entire community. It serves cellular, circuit, developmental, systems neuroscience, bridging from cellular to neuroimaging scale, serving the disease community so that we can all understand disease at a finer level of resolution, and serving other major NIH initiatives, such as the Connects program, which will be moving from cells to circuitry, the armamentarium program that takes this information and can use it to derive tools to be able to selectively target cell populations and to integrate with the rest of the community, such as HubMap and Human Cell Atlas and many others, to integrate the brain into the whole body. As I think all of you know, the previous stage of the cell census projects, or the Brain Initiative Cell Census Network, or BICCN, had several really flagship achievements. On the mouse side, really remarkable set of whole brain comprehensive cell atlases with both single cell transcriptomics and also with spatial methods, MERFISH in particular, to create a comprehensive view of the cellular makeup of the brain. And as all of you here appreciate very well, this really illustrated the complexity of the system we're dealing with more than 5,000 clusters that can be identified. And in most cases where we go and look with other methods, these correspond with other phenotypes of cells as well. In parallel, we also achieved the first draft of a human brain cell atlas that already got to about 3,000 clusters by sampling a defined set of regions. So this was really setting the framework and the stage for BICAN. And in BICAN, we've taken this to an entirely different level by trying to create comprehensive human and non-human primate cell atlases to do this in the adult and across development, and to do this across populations to understand individual variation. This includes a whole suite of tools and approaches to do this, including trying to derive common coordinate frameworks for all of these species, to have harmonized nomenclature for structural ontologies across the species, to have the components of cell atlases, so single cell transcriptomics and epigenomics, spatial transcriptomics, cellular phenotyping, to derive these robust cellular taxonomies that can then be turned into these foundational consensus atlases and references. Along with this come a whole suite of tools for data visualization and analysis, and a knowledge portal for aggregating information. So I'll describe briefly some of these components here today, but this is really what we're trying to do for the entire brain now. As Zeng began to mention, this really involved a lot more than we had done before, with the goal of a set of data products and some nice papers, but to really centralize things and to try to create this community reference. This involved a lot more standardization on everything from tissue access and human brain access in particular, to anatomical sampling, to standards and standardized data pipelines for producing this, for aggregating and documenting all this information through, for example, our neuroanatomy anchored information management system, and released through the knowledge platform and also the data archives. Conceptually, we've really been following the conceptual framework of the genome. And I think that this still really holds for a lot of what we're doing here, that we're now at a place where we're all really describing the same system, and it's quite robust with these methodologies. And we can think of this now as aiming for a more formalized set of cell references in the spirit of the genome. So for example, this would have robust cell taxonomies, both within species and harmonized across species. We would have data-driven ontologies with a nomenclature system similar to the genome, but with aliases for different names. For example, tools to visualize and mine the data, tools to map against the data. This is really important for people to use it, to be able to map and transfer labels onto their own data. We have tools like Map My Cells to do this. Rahul has generated tools to do this as well, I think is in the audience, and knowledge bases to aggregate all of this information. Our own project in the consortium is in the adult brain is called the Human and Mammalian Brain Atlas, or HMBA. This is really the broadest project trying to cover the entire anatomy of the human and macaque and marmoset brain. We've approached this in a highly parallel fashion across species to try to get really comparable coverage so that we can adequately map homologous cell types or understand species specializations. This involves single-cell genomics coverage, now bringing spatial transcriptomics across the brain, phenotyping of cells with techniques like patch-seq, and producing a series of products. And in particular, I want to call out that 3 core types of products we're trying to produce from this are the cross-species consensus cell taxonomy, the spatial map with spatial transcriptomics, and a common coordinate framework in which all of these can be mapped that can bridge scales also, and bridge from neuroimaging to cellular molecular scales. As we did in BICCN, we focused initially on one part of the brain. And in this case, we decided to go subcortically and focus on the components of the basal ganglia circuitry. In part, this is because of its high disease relevance. Of course, this is involved in a whole range of different disorders, motor disorders like Parkinson's, Huntington's, also more cognitive disorders and addiction as well. And by bringing the whole strength of the consortium together, we could really explore this structure in great detail to get at cellular diversity, species conservation, or perhaps divergence in certain places, developmental mechanisms, regulatory mechanisms, and even push this towards disease studies or gene therapy applications with the genetic tools as well. This has been extremely successful. We've now produced a cross-species harmonized cell taxonomy of the basal ganglia, And I have to say, when we do it this way, the degree of conservation down to a certain level in the taxonomy, at least, is really astonishing. Across the primates, it's very highly conserved. But we can define all of these cell populations very robustly. We've now created taxonomy to match that, and with nomenclature that actually works across species, so they can't use names of markers, for example, that aren't conserved. We have spatial mapping across all 3 species and cellular phenotyping in non-human primate of these. We've used this to create formal cell ontologies and to put this into a brain knowledge platform, and we have tools through MapMyCells for the community to map against this and transfer labels onto their own data sets. So this is really sort of the epitome of what we're trying to do across the whole brain. Focus on the basal ganglia was intended to be a paradigm setter for us to think about how best to do this, how to aggregate and integrate this information to produce this. In the meantime, we've already generated basically all the data for everything subcortical in the entire brain across species. And so in the theme of this meeting, the data production far outpaces the data interpretation, but we're in a great place now to be able to move forward towards the rest of the brain. A couple other things I wanted to mention. The first is that if we want to get adoption across the community, we really need to engage the community on this annotation. And we began to do this for the basal ganglia. We collaborated with some terrific colleagues from the Human Cell Atlas that produced a tool called the Cell Annotation Platform, or CAP. This is an online tool that allows us to put a cellular data set and taxonomy into it and allow community input into this. We held a jamboree for this to try to get input from a series of domain experts, and really help to refine this taxonomy and also the nomenclature system around it. And this is something I think that we'll need to use for the rest of the brain as well. Now provides a mechanism for actually aggregating input from the brain, which is really important. All of this is leading to a set of really, I hope, impactful tools. And the whole ecosystem that's been created by BICAN is producing a lot of value that I think will have high impact here for people being able to access this data through the knowledge platform, through a series of other tools that allow access to different kinds of data, and in aggregate are a really powerful way of exposing these data and the cell ontologies that we're hoping the community will adopt more broadly. As Young mentioned, we're now sort of scientifically in a great place as well with the first paper package coming out of this. As we've done in the past, we've tried to coordinate activities across groups that are focused on this same theme. So the package contains a whole series of papers that are describing the basal ganglia with all the different modalities that we have to bring to bear here, as well as the many infrastructure components of BICAN as a whole. And a series of related atlasing studies in other structures as well. And the thing I really want to highlight from this, besides just the incredible breadth of research and productivity from the group, is that we've really tried to have all of the groups mapped to this same reference. And that's key. We have not accomplished that in any of the previous packages, that we want to show adoption of this reference ourselves so that we could foster adoption by the community. And we really want this effort to become the community standard rather than every paper redefining the cellular landscape anew. We're at a place now where we've described this well enough that this can become the standard, and we hope that the whole community and many of you in the audience here that may not have been part of this will begin to adopt this and help to iteratively refine it rather than trying to create it de novo in each study. So finally, this has set the stage towards the future. I think we're in a very, very different place here now. The basal ganglia was intended as a paradigm setter for how to go about doing this for the whole brain. There are a whole series of other problems, and Hongkui will get into some of these, of data integration across, even within the same species, different atlases, but now between species as well. And I just sort of lay out that I think we have so much data now that it's really changed the approach to doing this, that we can begin with cross-species integration from the outset to get a coarse, whole brain, cross-species ontology or taxonomy, and then begin to break this out into parts for much more detailed annotation. And the current trajectory is to do this whole brain integration now, and that will be done actually in the next months, and then to part out the brain into structures where we'll have domain experts helping on the annotation of those structures with the CAP tool and other things to really try to get to this highly refined set of structural regional atlases that eventually will then aggregate to have a whole brain atlas. And so hopefully this will help to set the stage for some of the future discussions in the meeting today. Just finally, I'd like to acknowledge that this is a huge consortium effort. Our own project already involves many institutes and many domain experts that have generously agreed to work together to try to create this foundational reference. And I think this has been really successful to bridge across scales, across modalities, and bring this whole thing together to try to create this reference, but also acknowledging that this is one project within the whole BICAN landscape, and really everyone is involved with this. And so this is a joint effort across many individuals that I think we're all trying to do this impactful thing for the community. And so with that, let me turn over to Hongkui.
Okay, thanks, Ed. If you can advance, slides for me, that'd be great.
Sure.
So I will follow up on Ed's great presentation, overview presentation, focusing on 3 future directions, which is really creating consensus integrated cell atlases, starting with integrating with the mouse at a whole brain scale, and then integrating across species, again at a whole brain scale, and finally talking about the generation of developing cell atlases across species as well. So in this first single slide, I'll introduce to you our recent effort at the Allen Institute, led by Changqiu C.K. Lee and Zeng Yao, in computationally integrate 2 major whole brain cell atlas taxonomy data sets that we published in 2023. One is from Allen Institute, we call it ABC Atlas, a single cell RNA-seq data across the entire brain. And another is a similarly large data set across the entire brain from Evan McCarskill's lab, single nucleus RNA-seq. So using a scaled up computational approach, we used the SEVI to integrate those 2 data sets together, as you can see here, creating a consensus or integrated single cell and single nucleus mouse whole brain atlas, now containing more than 7.6 million cells or nuclei after stringent QC, and a definition of new set of classes, subclasses, supertypes, and clusters. The 2 data sets, as shown in the UMAP here, have been integrated really well, even though there are still some differences in single cell and single nucleus data. We are able to match them all the way down to cluster level. And we have now reached a higher level number of clusters, 6,700 now, compared to about 5,000 that we defined before. And this increased cluster number is mainly because the complementarity of the 2 different data sets. The single, with some subclasses or clusters depleted in one data set, but present in the other. So by joining the 2 together, it really enhances and gives us a much fuller picture of the whole mouse brain atlas. For example, in the single cell RNA-seq data from Allen Institute, we undersampled very large neurons in the medulla and pons areas that complemented by the single nucleus data, whereas in the single nucleus data, hypothalamus is significantly undersampled. That's now also complemented by the single cell. So this shows now this is a new integrated platform that we are presenting to the community, and it has already been released at SFN last year. The entire data set, along with cluster annotation, is now becoming available. Next slide, please. And then moving from here, we have also been developing computational tools to achieve whole brain scale cross-species integration. In this current version, which is the integration between the whole mouse brain data set from Allen Institute and a whole human data set from Stan Lenassen's group in collaboration with Ed Lein at Allen. Altogether with 8 million, 7 million cells or nuclei, we also used an iterative, integrated SCVI approach to co-embed the two data sets and achieve a subclass level integration that has worked quite well. Here in this slide, we're just showing two example neighborhoods, which were known to be extremely challenging to resolve in previous attempts. This midbrain, hindbrain glutamatergic and midbrain, hindbrain GABAergic neighborhoods, previously in the Lenasen or Salati papers were described as a big splatter. And now we are able to resolve here, as you can see, as well as really excellent cross-species alignment at the individual cell level, with only a few subclasses, as you can see here, as shown in the circled here, that show species preference or incomplete integration, at this kind of global scale, but we already know their correspondence very well. So if you can move to the next slide. And then here summarizes the integration of the whole human and whole mouse data sets at subclass level that allows us to transfer all the subclass labels. So by the way, this is still only the Allen brain cell ABC atlas, single cell taxonomy with 334 subclasses, but we're now able to transfer all of them into the human data and achieve a really high degree of alignment as shown here. There are a few gaps in the human data because we think that cells and those subclasses were undersampled in the Salati dataset. But overall, very good level of consistency as shown by the random forest mapping probability here, at all subclass level. And finally, another way of demonstrating in the next slide, it shows that after subclass level alignment across species, we're able to also examine conserved and divergent gene expression. And we find that transcription factors actually show very high degree of alignment, consistency, conservation between mouse and human, once all the subclasses are aligned well. So in this particular heat map, every transcription factor is shown in the column, and every the expression of all the transcription factors across species for each subclass is shown in a row. And they are arranged according to the 34 classes that we defined in the mouse. And as you can see here, really striking level of conservancy of transcription factors, genes across the two, very distant species. So demonstrating the a good quality of alignment as well. And we can do this for other gene families as well. And many of the other gene families are less well conserved as transcription factors, again, suggesting that its transcription factors are really driving forces in defining cell types, even in the adult brain. And finally, in the next slide, I'll talk about our, consortium-wide recent progress in generating developmental cell atlases across mammalian brains, from mouse to marmoset to macaque and the human. And here just shows the publication package that was just published in November 2025. And this project is really a bridge from BICCN to BICAM. In this package, there are 9 papers published in Nature, 2 papers in Nature Neuroscience, and 2 more papers in Nature Communications. Collectively, begin to paint, this kind of developmental dynamics of the cell types in different parts of the brain and across species. And this particular diagram, as we laid out in the perspective paper associated with package, shows the alignment of developmental timelines and key developmental events across the species. And our ultimate goal from the consortium is to build high spatial and temporal resolution cell atlases across the entire developmental periods, prenatal and postnatal across species, and so that we can align them as well, just like what we have been doing for the adult brain, to understand cross-species conservation and divergence in terms of the emergence of cellular diversity and circuit specificity. And then finally, the last slide. Here is a summary, again, using a diagram from the perspective to really summarize the collective work from the BICAN consortium with major investigators from the different grant award groups showing here, but also many more other participating investigators that I'm not able to list here. Our collective goal here is to use the multi-model genomics approaches, including RNA-seq, ATAC-seq, and other epigenomic approaches, as well as spatial transcriptomics and lineage tracing, different methods of lineage tracing, to derive the cell type atlases and achieve cross-developmental description and cross-species alignment, and also understanding the spatial distribution of the cells and how the different cell types migrate throughout the development. So creating this four-dimensional spatial temporal coordinated atlas. And with that, understanding the molecular mechanisms as well, including the lineage and trajectory relationships of cell types across time, cell types and cell states.
Could you repeat that last sentence or two?
Okay, yeah, I just said that I don't know why I was cut off, but yeah, just say we're concluding our presentation, and I don't know if we have time for questions or not.
Yeah, we're out of time, but I think we'll answer one that came in. So someone had a question about the size of the microfluidics of the 10x genomics chips. They say 10x cell size recommendation is 35 microns, but neurons are pretty big. I'm wondering, this is a kind of technical detail. What were the size of the microfluidics of the 10x genomics chips used?
Larger neurons would be depleted in the microfluidic condition.
Yeah,
So
on the size. I think that's what this person was asking.
I think gigantic neurons are usually found in primate brains, especially human brains. But in a human brain, we usually profile nuclei rather than whole cells. So we can kind of get away that problem to some extent. But we understand that gigantic neurons are still a challenge to profile.
Matt Thornton has a question, and then we'll move on to the next keynote.
My name's Matt, and I work at USC, and I'm a research specialist. And sometimes I do bioinformatic analysis. And we have had researchers where they were wanting to look at a specific type of neuron in a mouse brain. And we also have a 10x genomics core where they process the 3' chromium chips. And what we noticed was that the chips kept getting clogged up when they would apply the neurons to the chips. And so I looked online, and the recommended cell size for 10x genomics is 35 microns, which is around blood size, like red blood cell size, white blood cell size, things like that. Neurons even regular neurons are hundreds of microns. And then if you have their hydrodynamic radius, because they have such a strange size, that's going to be much, much larger than the microfluidics of the 3' chromium chips. And so what I was wondering is, I mean, you do have a lot of correlation between the different experimental types, but I was wondering if that's all just to cover up for this bad representation of neurons in single cell RNA sequencing.
Maybe just very briefly, yes, I think that is a challenge we should be aware of. We think that a single nucleus RNA-seq will alleviate this problem to very much extent, but we should be aware of this issue.
Thank you very much. I really appreciate it. I was just wondering, because we had tried to integrate the data, and we didn't get any contribution from neurons, and so I was wondering if that was a more common state than I realized, and I guess it is.
Yeah, very large neurons are very difficult to profile.